Hi, good morning. This is Kavita Kundra, and now we are actually coming up with the fourth part of carbohydrate metabolism of a book, MSc DFSM Biochemistry. So, this is regulation of gluconeogenesis. Gluconeogenesis and glycolysis are reciprocal. All factors that increase gluconeogenesis decreases glycolysis. That means whatever factors increases the gluconeogenesis, those factors will decrease glycolysis. Gluconeogenesis means new glucose formation. Glycolysis means glucose breakdown. So it's obvious the same thing. The key enzyme of gluconeogenesis include pyruvate carboxylase, phosphophenol pyruvate carboxylase, and glucose 6 phosphatase. Important aspects related to regulation include the hormone glucagon and glucocorticoid that are secreted during starvation stimulate glucose 6 phosphatase to enhance gluconeogenesis. During starvation, the increased level of glucagon also stimulate the enzyme phosphoenol pyruvate carboxy kinase, increase gluconeogenesis. During starvation, more acetyl coenzyme produced that activate the enzyme pyruvate carboxylase and form oxaloacetate and enhance gluconeogenesis. The released glucagon decreases fructose 2 by bisphosphate and stimulate gluconeogenesis and in turn activate fructose 1,6 bisphosphatase. High carbohydrate diet increases insulin or glucagon ratio and minimize the gluconeogenic mechanism by reducing enzyme activity. ATP and citrate are the activators of fructose 1,6 bisphosphate and gluconeogenesis increase. High level of AMP inhibits fructose 1,6 bisphosphate and gluconeogenesis is reduced. Increased ATP inhibits pyruvate carboxylase and reduces gluconeogenesis. The hormone glucagon, epinephrine, glucocorticoid synthesize pyruvate carboxylase and increases gluconeogenesis. But the insulin depresses the enzyme pyruvate carboxylase and reduces gluconeogenesis. What are the regulatory roles of fructose 2,6 bisphosphate? It is most potent allosteric effector of phosphofructokinase. 1. Inhibitor of fructose 1,6 bisphosphatase in liver. Second, it relieves inhibition of phosphofructokinase 1 by ATP and increase affinity for fructose 6 phosphate. Third, it inhibits fructose 1,6 bisphosphatase by increasing the Km of fructose 1,6 bisphosphate. Fructose 2,6 bisphosphate is formed from fructose 6 phosphate by enzyme phosphofructokinase 2. The same enzyme also causes its breakdown by possessing fructose 2,6 bisphosphatase. Activity is controlled by fructose 6 phosphate. When glucose is less, the glucagon stimulates the production of CAMP which inactivates phosphofructokinase 2 and fructose 2,6-bisphosphatase. When glucose is abundant, fructose 2,6-bisphosphate stimulate glycolysis by activating phosphofructokinase and inhibiting fructose 1,6-bisphosphatase. In glucose shortage, gluconeogenesis is stimulated by decrease in the concentration of fructose 2,6-bisphosphate that deactivate phosphofructokinase 1. This mechanism also shows that glucagon stimulation of glycogenolysis in liver result in glucose release. Metabolism of glycogen. The glycogen is highly branched and large polymer of glucose molecule linked along its main line by alpha-1,4 glycosidic linkage. Branches arises by alpha-1,6 glycosidic bonds. The shortage form of glucose is glycogen, and glycogen stores in liver and muscle. Although glycogen is higher in liver, the skeletal mass also stores greater amount of glycogen. When a body goes under exercise, muscle glycogen gets depleted and muscle glycogen act as source of hexose units for glycolysis. Liver glycogen is responsible for storage and export of hexose unit for maintaining blood glucose. After 12 to 18 hours of fasting, the liver glycogen is completely depleted. After a meal, synthesis of glycogen and liver is initiated. This process is called glycogenesis. This process prevents excessive rise in blood glucose level but helps to store glycogen for future use. In the liver, glycogen is metabolized to glucose and released into the circulation when a person fasts. In muscle, glycogen cannot be converted to glucose. It can still be used for obtaining energy during muscle contraction. This breakdown of glycogen in liver and muscle is called glycogenolysis. Glycogen synthesis and glycogenolysis occurs in different pathways. Glycogenesis. The process of glycogen in liver and muscle is called glycogenesis process. Glucose is phosphorylated to glucose 6-phosphate by hexokinase in presence of ATP and magnesium ions. Glucokinase remove glucose from blood following a meal. Phosphoglucomutase convert glucose 6-phosphate to glucose 1-phosphate, and this is reversible reaction. That means glucose 1-phosphate can again be converted into glucose 6-phosphate. Glucose 1-phosphate reacts with uridine triphosphate to form UDPG, uridine diphosphate glucose. With the help of enzyme UDPG, pyrophosphorylase and PPI molecule gets converted to two inorganic pi molecule. The synthesis of new glycogen requires primer and alpha glucosyl residues from UDP glucose. The residues are successfully transferred to C4 terminus of an existing glycogen chain in alpha 1,4 glycosidic linkage. This process is continued till 10 to 12 molecules have added. This reaction is catalyzed by glycogen synthase. After the chain is lengthened up to 11 glucose molecule, the enzyme trans-glucosidase transfer a part minimum of 6 glucose residue to the neighboring chain to form 1,6 linkages by adding 1,4 glucoside units and further branching. Regulation of glycogenesis. The main key enzyme is glycogen synthase. It is activated by insulin and glucose and inhibited by cAMP. Glycogen synthase exists in two forms. One is phosphorylated D form, that is an inactive form, and dephosphorylated form, that is an active form. D form is allosteric enzyme that is activated by high concentration of 5-AMP, and D doesn't require 5-AMP. Protein kinase catalyze interconversion of D and I. The level of CAMP is in turn regulated by adenylate cyclase enzyme, which is activated by glucagon and epinephrine. Glycogenolysis. Glycogenolysis is breakdown of glycogen. Glycogen is broken down in the liver and muscle, catalyzed by enzyme glycogen phosphorylase. Inorganic phosphate, that is pi, is used for lysis and is called phosphorylasis. Phosphorylase acts upon alpha-1 glucoside residue, continues till 4 glucose residue remain on either side of alpha-1 6 branch, then debranching enzyme amylo alpha-1 6 glucosidase causes hydrolytic splitting of alpha-1 6 linkage. Here free glucose is formed. Since alpha-1 6 linkage are very few compared to alpha-1 4 linkage, the major end product of glucogenolysis is glucose with small amount of glucose 1 phosphate. By combined action of both enzymes, glycogen is catabolized. The reversible reaction of phosphoglucomutase causes the conversion of glucose 1 phosphate to glucose 6 phosphate. In liver and kidney, glucose 6 phosphatase act on glucose 6 phosphate to release free glucose from the cell to extracellular compartment. Kidney glucose 6 phosphatase act on glucose 6 phosphate to release free glucose. Glucose phosphorylase is dimeric enzyme with 2 polypeptide that utilize pyridoxal phosphate as prosthetic group. Different isozymes of glycogen phosphorylase are present in different tissues. Glucagon stimulated CAMP level activate phosphorylase from liver. Muscle phosphorylase is activated by epinephrine via CAMP. In lysozyme, alpha-1 4 glucosidase is involved in debranching. Regulation of glycogenolysis. Glycogen phosphorylation exists in 2 states: phosphorylase A, that is active state, and phosphorylase B, that is inactive state. Catecholamines, epinephrine and norepinephrine, and glucagon causes the increase in CAMP level in the cell, and the CAMP activates protein kinase that stimulate key enzyme phosphorylase for glycogenolysis. After muscle contraction, glycogenolysis is highly increased in muscle by rapid activation of phosphorylase due to activation of phosphorylase kinase by calcium ion. Calmodulin causes further activation of phosphorylase kinase for glycogenolysis. Now, as we have seen that regulation of glucose phosphorylation is complex, its regulation ensures that glucose remains stored as glycogen until it is metabolized from the liver for maintaining blood glucose homeostasis or to supply energy to the muscle cell. Phosphorylase kinase is responsible for catalyzation, which is activated either by calcium or phosphorylation. The cascade is initiated by binding the hormone to its specific receptor. Hormone binding activates adenylyl cyclase. Adenylyl cyclase produces cyclic AMP that activates protein kinase. Protein kinase catalyze the phosphorylation of phosphorylase kinase, converting it from its inactive form to active form. Phosphorylase kinase catalyzes the phosphorylation of glycogen phosphorylase B to generate glycogen phosphorylase to form glycogen degradation is triggered. Thank you so much. I hope you enjoyed this chapter, and this is the topic where we have not completed yet carbohydrate metabolism. This is a big chapter and you have to go through very carefully because there are lots of terms and lots of reaction. It's a reaction based. The person who are very good in chemistry, they can do it very well, but if the person is who is not expert in chemistry, who has little difficulty to understand the chemistry reactions and all, so please go through it very thoroughly again and again till it is done in your mind. So we'll take your leave, and now we will be coming again with the next part of this topic. I hope we will be completing this chapter soon. Thank you so much. Stay safe, stay protected. Bye bye.